Hola, muy buenas tardes. Tengan ustedes un feliz día. Los saluda cordialmente la RELAGH, la Red Latinoamericana de Genética Humana. Y a continuación queremos darle la bienvenida a todos ustedes a esta conferencia que nos la va a presentar la doctora Mayana Zatz. Mi nombre es Harvey Velasco y con gusto invitamos a la doctora Mayana Zatz a que esté con nosotros. La doctora Mayana Zatz es profesora de Genética Médica y Humana, es directora del Centro de Investigación en el Genoma, enfocado específicamente en temas de envejecimiento y genética, metagenómica y genómica en la Universidad de São Paulo, graduada de Biología de la USP con un PH en Genética Médica y Humana, también de la USP, y una estancia postdoctoral en Genética Médica de la Universidad de California. Es miembro de la Academia Brasilera de Ciencias y de la Academia de Ciencias para el Mundo Desarrollado. Fue fundadora y presidenta de la Asociación de Distrofia Muscular de Brasil, y su investigación y sus investigaciones en genética humana y médica se han enfocado principalmente en stem cells, envejecimiento, desórdenes musculares y otros aspectos también muy relevantes, como el caso de Zika. Doctora Mayana, es un placer tenerla acá con nosotros. Obrigado por su presencia.
Muchísimas gracias. Eu vou falar em inglês. I would like to thank you very much for the organizer to invite me to talk a little bit about our research, about the future of medicine, and what we have been doing in our center. So what were the revolutions of the last 25 years? First, the cloning of the sheep Dolly in 1986, the Human Genome Project that started in 1990, the iPS-induced pluripotent stem cells, and then the gene editing in 2012. How these breakthroughs will and are changing medicine. The first example is the DNA and RNA vaccines against SARS-CoV-2 that were developed so fast. So in the beginning, we all started as a fertilized egg, and all our genome was inherited from our parents is inside this fertilized egg. And first we have the pluripotent stem cells, which are in the embryonic cells that have the potential to form a entire individual if we insert it in uterus. But then after 5 days, you have already the blastocyst, and then you have the first differentiation. The outside cells will differentiate in placenta and embryonic membranes, while the inside cells will give rise to the pluripotent stem cells. And then the stem cells differentiation continued. You have the different cell lines, and then the formation of the organs. So the big question we wanted to address was how cell fates are determined. What we know is once differentiate in a specific tissue, all daughter cells maintain the same characteristics. And we knew that the secrets were in embryonic stem cells, and that's why people, investigators all over the world wanted to study the embryonic stem cells. And then we had the first breakthrough, which was the sheep that was cloned. Dolly was the sheep that was cloned by Kate Campbell and Ian Wilmot. Kate came to visit us in our center in Brazil. And when the first sheep was cloned, there were a lot of ethical issues that were raised. They were saying, well, they are playing God, they will clone humans, who will be cloned, who decided who should be cloned? But nothing of this happened. What was the real Dolly's revolution is that she demonstrated for the first time that it was possible to reprogram a differential mammalian cell to the embryonic stage and to differentiate in all cell types. And if introducing a surrogate mother uterus to generate a cloned animal. And then we had the Human Genome Project. The big question was, what is the genetic versus environment role in a human characteristics? Because we have some characteristics that we know that depends only on environment. For example, the language that we speak. while others depends only on genetics, for example, our blood groups. But most of our characteristics depend on interaction between genes and environment. And then in order to know what were this role of the genetics, the Human Genome Project was launched in 1990. And the bet was that we would finish this in 2005, and that the estimates were that we had between 8,000 to 150,000 genes. And what was found in 2003, the Human Genome Project was announced, the end of the Human Genome Project. And at the same time, it was celebrating the 50 years of DNA double helix discovery. And this was Francis Collins, who was directing the Human Genome Project. And he was very happy and saying, the human genome sequence is finished, we did it. And then the big question was, how many genes do we have? Well, to our disappointment, we found out that we have only 20 to 21,000 genes. Just to have an idea, C. elegans, which is a small worm of 1 millimeter only, has 20,000 genes. And the tomato has more than 30,000 genes. So we lost even from the tomato. But obviously, we found out that they are much more complex than we thought. So the big challenge is to understand what is the gene's product, how their expression is regulated. And this is the functional genome. So we know that the human genome has 32 billion base pair, but only 2% of them codify protein genes. What is the role of the remaining genome, the non-coding genome? And then there was several publications, one in Nature in 2020, which is the ENCODE project, the Encyclopedia of Coding Elements. And they've shown that they have different transcripts within each gene, and the gene expression depends on millions of regulatory elements, enhancers, repressors, silencers, and promoters. And that's what explains the variability of our genome. So our approach in our center to the Human Genome Project was to identify and perform functional genes in genes causing genetic disorders. Because when you identify a disease-causing genes, this allows to understand their function. And then the next breakthrough was the discovery of the induced pluripotent stem cells by Shinya Yamanaka, who received the Nobel Prize in 2012 because of this discovery. And he showed for the first time it was possible to reprogram mature cells to the pluripotent stage. He started with fibroblasts of mouse and then from humans, and showed that activating 4 transcription factors that were called the Yamanaka factors, you had the iPS cells who had the potential to differentiate in all cell lines. So how this works, he started with human fibroblast cells, but now we can do this with blood. And when you activate these 4 transcription factors, you can reprogram cells first to the stage of iPS, and then you can differentiate these cells in all cell lines. For example, hematopoietic, pancreatic, neural cells, adipocyte, cardiomyocyte, all cells that you want to study. What are the advantages of the iPS stem cells? Well, you can be anything you want when you grow up, says the stem cell parental advice. So you can establish cell lines from patients with different mutations and perform functional studies. You can investigate why a mutation affects a specific tissue and not another. You can compare patients with the same mutations and different phenotypes. You can correct or create mutations and test different drugs in these cell lines. For example, we can derive neurons from centenarians, from erythroblasts, and understand and study how the neurons function on these healthy centenarians. And then we had another breakthrough, which was the new tools of genetic engineering, the CRISPR XVIII, which was discovered by Jenny Verdun and Emmanuel Charpentier in 2012, and also who gave them the Nobel Prize in 2020. So how is this DNA editing, the CRISPR-Cas9 technology? It's like a program find and replace function. So you have a cell that is transfected with an enzyme containing a guide molecule and a healthy DNA copy and DNA cutting enzyme, which in the case is Cas9. Then you have a guide molecule, which will find the place in the DNA that you want to correct. Then the enzyme cuts this DNA that needs to be corrected, and the defective gene is replaced by a healthy copy. So how these breakthroughs will and are changing medicine? And you can give several examples. You'll have in gene editing and gene therapy, precision medicine, that we call it P4, the identification of protective genes and what is their mechanism, xenotransplantation, regenerative medicine and tissue engineering, resistance to infectious disease, for example, SARS-CoV-2, and I'm going to show you also how to transform your enemy in an ally. The gene editing CRISPR-Cas9 is already in clinical trials in several conditions, like some forms of cancer, hematological conditions like thalassemia and sickle cell anemia, some forms of inherited blindness, Huntington chorea, cystic fibrosis, just to give some examples. And then what will be the future of medicine, a medicine that we call P4? It will be predictive, preventive, personalized, and participative. Predictive, how this will be predictive? Well, for a long time, we know already through the genetic studies that you can know if you have a Mendelian disorder, for example, inherited disorders, if a young person who did not develop the disorder yet has the pathogenic mutation. But there is an international consensus against testing young children for late onset disorders without available treatment. For example, Huntington chorea or cerebral ataxias. But another approach is what we call polygenic risk scores, which will estimate the risk that you'll develop complex disorders, such like diabetes, cardiopathy, hypertension, or cancer later in life. These studies are still ongoing, but certainly in the future, you can have more accurate estimates. So if you know that you are at high risk, you can take preventive measures. And then you'll have the personalized medicine. Interestingly, when the Human Genome Project was finished, it was shown that there is no superior race, which are all the same or equally different, but that ethnic or genomic difference can be very important in our response to drugs. So how will be the personalized or precision medicine? Now, when you take a medicine, we call it one size fits all, because you don't know if a new medicine will benefit you, show no benefit or adverse effects. And then when you have the personalized medicine, you'll take drugs according to your genetic background, the biomarker, genetic biomarkers, and you'll know exactly what drug will be good for you and in what amount. So you'll have no adverse effects. And then you'll have the participative medicine. So you're going to discuss with your doctor what is the best approach for your own health. So what are we investigating now in our center? Well, we have studied more than 100,000 families with different genetic disorders. And since the beginning, Oswaldo Frota Pessoa, who started the medical genetics in our centers, he always said there is a two-way road, patients originating new research and new research helping patients. So we have identified new genes or variants that can be very important for precision medicine. And so far, we have identified 40 novel genes for neuromuscular disorders, autism, intellectual deficiency, spastic paraplegia, craniofacial disorder, and skeletal dysplasia, among others. And we are now focusing on functional studies to understand what exactly they do and how we can correct the disease that is caused by these genes. Then about next generation sequencing. Just to remember, the Human Genome Project took 13 years to be finished at a cost of $3 billion. And now we have panels that can study more than 6,000 genes simultaneously. We have 94 for cancer predisposition, 600 for neuromuscular disorders, 110 for autism, just to give you some examples. But the big question is how to interpret variants pathogenicity when you have a new variant that you find in a patient. So in order to try to address this question, in 2008, we started what we call the 80+ project, which was, we started to collect the DNA and clinical data of healthy octogenarians. And the idea was to serve as a data bank of the Brazilian population to interpret variants pathogenicity and the search for protective genes and mechanisms responsible for healthy aging. The advantage of our population is that it is highly admixed. This is just an example of the people that work in our center. And then there was another project that was going on that's called SABI. It was directed by Maria Lúcia Lebrão and Yenda Duarte, where they had a follow-up of a cohort of people from São Paulo city, older than 60, that have been followed since 2000. So we joined together the two projects, the 80+ and this project, and we did genomic studies in these people that had all other clinical studies already being performed. And we are now publishing in Nature Communication the whole genome sequencing of over almost 1,200 elderly people from São Paulo, Brazil. And very interestingly, we started this more than 10 years ago, but recently the Cell, in the journal Cell, they showed that there was a missing diversity in human genetic studies. And they showed that the majority of studies of genetic association with disease have been performed in European, and that this has important implication for risk prediction of disease in global population. So they justify the need to study more diverse populations like ours, for example. And then interestingly, when you compare the big data genomic study, there are 600,000 in UK. There is a aim to have 1 million genomes in USA. And what can we say about only 1,200 in Brazil? Well, we had big results because we found in our cohort about 2 million variants that were not reported before in the European databanks. For example, we found 10 subjects that had pathogenic mutations for 6 cancer genes, 2 for breast cancer and other for other types of cancer. And we recontacted 8 of these elderly, 2 reported cancer, but 6 of them reported no history of cancer, including a lady who was 95 years old. She had a pathogenic mutation in the BRCA gene, which is responsible for an inherited form of breast cancer, and she never had any kind of cancer nor her offspring, showing that the same mutation can express differently according to different genetic background. Could it be because they have protective variants or different lifestyle? But it shows the importance of studying different populations. Well, and how genome sequencing is and will contributing to novel treatments. An example was with a spinal muscular atrophy, which is caused by motor neuron death. All the patients have a deletion of the FSM gene that is called survival motor neurons, but it was shown already before 10 or 20 years ago that patients with the same deletion could have a very severe form and an intermediate form and an adult form with normal survival. And what was found is that this could be explained because they had a variable number of SMN2 copies, which are a similar gene, but has only 10% of expression. And then if you increase the expression of each copy, you could have a better phenotype. So with this information, so this is just to show it, this is the SMN gene and the SMN2 that produce a low protein level, only each copy only 10%. So you know that if you have 4 copies, you can have an almost normal phenotype. So the company Spinraza used this information and to do antisense oligonucleotides and to increase the amount of SMN protein levels. And this is a treatment that's already ongoing. And another one, another approach is through gene therapy, where you insert a copy of a normal gene in a virus and which infect the body with this virus. It was approved by FDA, but the big problem is that the cost is over $2 million per treatment. So we have to fight to have these treatments affordable for all patients. And then we have been looking for searching protective genes variants in asymptomatic patients carrying pathogenic mutations and also in centenarians. So today there are about 500,000 centenarians around the world. What are their secrets? Diet, protective genes, lifestyle. There is a very interesting study by Nir Barzilai, who studied centenarians, and he showed that 60% of men and 30% of women smoked for long periods of their life. Almost 50% of them were overweight, and less than 50% did physical activity. So they probably have protective genetic variants. And he showed, for example, 4 brothers, the Can brothers, who had a variant associated with very low levels of LDL, which is the good cholesterol. And this could be a protection against Alzheimer's and increased longevity. But it was very interesting, but because he showed an interview that he had with one of the sisters who was 106 at the time, and she confessed to him that she had smoked all her life. And he said, well, your doctor didn't tell you that you shouldn't smoke. And she replied, yes, I had 4 doctors that always told me that, but unfortunately, all of them already died. So what was observed in the centenarians in Okinawa? They had rare HLA variants, and which was related to the increased risk of inflammatory and autoimmune disease. And then there was a study of genomes of supercentenarians who lived more than 114 years. And when they looked at their genomes, they had genes that are comparable to common genomes, like Alzheimer's, cancer, cardiac diseases. So again, it is believed that they have protective genes. So you have these protective variants that you can identify as protective variants. The next question is, what is their function? How to use this information to protect everyone that was not born with these variants? And what other approach could be planned in the future? So just for an example, we have some candidates of variants that are, should explain this longevity. like the STP-TP that is involved in the regulation of high-density lipoproteins, apolipoprotein C3, which is a component of very low lipoprotein, adiponectin or MTP, just to give you some examples. So how could you use this information? With CRISPR-Cas9 technology, you could correct or edit defective genes or genes that are responsible for rapid aging. You could transform common genes into protective genes. You could increase the expression of these protective variants. So the first thing, you tried all this approach in cell lines. If it works, you work in mice, and then this could be transformed in gene therapy. You could also inject a copy of the protective variant in a cell. You have the cell with a gene, you introduce the protective variant in the cell, and the cell function is rejuvenated. What can we learn also from rare exceptional patients? So we have been working a lot in our center with Duchenne muscular dystrophy, which is caused by mutation in the dystrophin gene. And the patients have this mutation that transform a normal muscle the muscle degenerates because he has no dystrophin. So this is a normal muscle. This is dystrophin around the cell. And here is a Duchenne patient who has no dystrophin in its muscle. And then we have been working with, there are dogs that are golden retriever muscular dystrophy who are born with this same mutation, and they have a very severe phenotype, very similar to the patients. 80% of them do not survive beyond age 2. And again, this is a normal muscle of a dog, a golden retriever dog, and a dog which has the mutation and no muscle dystrophin. And we were able to study 2 dogs who had the same mutation, no muscular dystrophin, and a normal lifespan. So this was Ringo, who lived for 11 years and had a almost normal muscle and performance, and his son, Souffler, who also had a very mild phenotype. So we published in 2015 a paper showing that you can have a normal life in dogs without muscular dystrophin, which was the first time this was reported in the literature. So just to show that they had no muscle dystrophin at all in their muscle with immunofluorescence and Western blot analysis. So the big question was, what is their protective mechanism? And we found out that they have an increased muscle expression of JAG1, which is 2 times higher in this escapers dog than in normal severely affected dogs. And this was confirmed by protein analysis. So finding like that in dogs can open new avenues of possible treatment. And it was published in Cell in 2015. We also have been working in regenerative medicine, and the question is, should we reconstruct organs or replace them with new ones? So just to have an idea, every 30 seconds, a patient dies worldwide in the waiting list for an organ transplantation. So we have been working in this, what we call the organs approach. This is Silvano Raia, who was the first, he was pioneer in the world to do the first liver transplantation among people that were alive. Around there are many, many thousand people that have been benefit with this transplantation. So one of our approach will be xenotransplantation with pig's organs. Pig's genes will be edited to avoid organs rejection when transplanted in humans. And this was already shown to be successful when hearts of pigs were transplanted in baboons, and they were able to live for several years without any problem. So this is our group working in our lab with this approach. It's a partnership where we have also a private company that is investing in this project. And what would be the approach? You have wild pigs. We took fibroblasts from these pigs, and we know that there are 3 genes that are responsible for acute rejection if transplanted to another animal or to humans. So we were able, with the CRISPR-Cas9 technology, to edit these 3 genes, and they have a knockdown all the 3 genes, so they are not expressed anymore. And then we did nucleus transfer, and the next step, it will be to implant this in a surrogate mother. And then you'll have the triple knockout pigs. So we have already the first edited embryo that was already successful, and we have we have our several blastocysts that we are going to insert in pigs, which will be the next step. And the idea is to start with kidneys. Just to have an idea, in 2018 in Brazil, we had more than 20,000 patients in the waiting list for kidney transplant, and 130,000 individuals undergoing hemodialysis. What we will start, why kidneys? Because you can implant the kidney underneath the skin, and if you have rejection, then you can take out this kidney and the patient can go back to hemodialysis. Another approach is what we call organs reconstruction. What is the idea? You can take a liver, for example, take out all the cells and leave out only the scaffold. And then you can recellularize this scaffold with patients-derived iPS liver cells. So how would this work? You take out skin or blood cells from a patient that needs a new liver. You reprogram these cells in induced pluripotent stem cells, and you differentiate in all cells that you need for a new liver. So you'll have a rebuilt organ that you can insert in the patient, and you wouldn't have rejection because you're using the cells of the own patient. When we have done this already with rat liver, here is the liver of the rat. Here, after we took out all the cells, and we were able to recellularize this rat liver with human cells, and this is what you see here. And we can do that also with 3D printers. So this is another project that is ongoing in our center. And then COVID-19 pandemic started. And the big question, how to explain the enormous variability in clinical case from severe lethal forms to asymptomatic cases. How to distinguish resistant individuals from asymptomatic non-exposed individuals. Could genetics explain an increased susceptibility or resistance against SARS-CoV-2? So in order to address this question, we decided to focus in asymptomatic exposed individuals. And how was this? We wanted to study discordant couples when one was affected and the partner was asymptomatic despite sharing the same bed during all the infection, and also nonagenarians and centenarians. And the third group were monozygote and dizygotic twins. So this is the group in our center that was working in this project. And we started to recruit volunteers through the media or friends. And to our surprise, we received more than 2,000 emails from more than 600 couples where they said one was symptomatic, while the spouse or partner was asymptomatic and seronegative. So the media was also very interested in this. And we had an email where people were writing. And this is Moniz and Mateus who were doing all the recruitment and lab work. So this is an example of a discordant couple living in the same house. He was sick for 2 weeks with high fever and all the symptoms of COVID, and the serum was IgG positive, while the wife who took care of him was absolutely asymptomatic and seronegative. And here an example of another family where this gentleman who was in his 70s was severely affected, and his wife and all the relatives were not affected, had nothing at all, including his mother, who was 98 at that time, all living in the same house. So we did whole exome sequencing in 86 discordant couple. The mean age was very similar among the infected and the resistant, but we found the first surprise were that there were much more women in the resistant group than men, 57 women and 29 men. And then we wanted to know what was going to happen with nonagenaries and centenaries, because we know that the age is a higher risk, represent a higher risk for people that are infected with SARS-CoV-2. And then we started to see announced in the newspaper people that were infected and survived. So every time there was an announce in the paper, we went after the patient, tried to collect samples from them. And we were able to collect samples from 100 nonagenarians and centenarians. 85 were nonagenarians, all cured, 2 were 99 years old, and 15 centenarians. And the centenarians mostly live in other Brazilian states, so they sent samples to us. There were 30 women, aged 100 to 114 years old, the oldest one, probably the oldest woman in the world to survive to COVID-19, and 2 men that were also centenarians. And we want also to do not only genetic studies, but functional studies. We are going to derive iPS cell lines from these centenarians and infect them in the lab with the coronavirus, and then try to understand why they are resistant. We don't know if the virus does not penetrate in their cells or activate cells that confer resistance. So this is another study that we are going to study in the next future. What about twins equally exposed? So we decided to compare monozygote versus dizygotic twins. And there were 7 cases in the newspapers of monozygotic twins that were infected and died days apart, which is very supportive of a genetic contribution to a severe outcome. We were able to collect samples from 10 pairs of twins. And when we compare monozygote and dizygote twins, we saw that 3 among 4 were concordant. When one was affected, the other also was. But among dizygotic twins, only 1 in 6 were concordant. And this supports also a genetic contribution. And our first paper was published in Frontier in Immunology. We found 2 genes, variants in 2 genes, MYC-A and MYC-B, which differed in symptomatic and as compared to asymptomatic partners. And we know that molecules codified by these variants modulate the NCAR, the natural killer activity, and which could contribute to susceptibility to COVID-19, downregulating the natural killer cells cytotoxic activity in infected individuals, but not in the asymptomatic partners. Well, we know that there are many more genes that are involved, but this was the first that we focused on, and this was our first result. And another very interesting thing that we showed in our study, that men are the main COVID-19 transmitters. And so when we compared men and women, we found out that in 943 cases, the men were the first to transmit it to women. And one possible explanation of this is that the viral load is 10 times greater in the saliva in men than in women, while there is no gender difference when we compare nasopharyngeal swabs between men and women. And this was published also in recently. And then finally, how did we transform our enemy in an ally? So Zika virus was first reported in Uganda in 1947, and the first cases were reported in America and in Brazil in May 2015. It is carried by Aedes aegypti, which also carry dengue and chikungunya. But the symptoms that relate to Zika virus are usually very mild. 80% or more of the people are absolutely asymptomatic. The big problem was the outcome of zero Zika virus infection during pregnancy, when it was shown that babies that were born with microcephaly and other malformations, the Zika congenital syndrome, when the mothers were infected during pregnancy. But my question was why most babies were not affected, because although each baby that was born was a tragedy, we knew that most of the women, pregnant women that were infected during pregnancy had normal babies. So could it be because of genetic variants, epigenetic mechanisms, viral load? And again, these were all the students and researchers that focus on this research. And one of our hypotheses that you could have a difference in neuroprogenital cell susceptibility for Zika virus infection. Just to remember that the neuroprogenital cells are the cells that will give rise to the brain in the fetus. So we derived NP cells from discordant twins when one was infected and the other were not, were dizygotic twins, and we infected them in the lab with Zika virus. And our hypothesis, if Zika syndrome would be related just to environment, monozygote and dizygotic twins should be very similar. But if it would be according to genes, monozygotic twins should be more alike than dizygotic twins. And indeed, we showed that in monozygotic twins, when the mother was infected, both were equally infected, but among dizygotic twins, most of them were discordant. And the study finished showing that congenital Zika syndrome is not a stochastic event. It depends on neuroprogenital cells increasing susceptibility and probably through epigenetic and oligogenic complex mechanism. And this was published in 2018. And the good news is that non-affected twins are developing normally. But what is the next story is how to transform your enemy in an ally, turning the bad guy into a good guy. So when I went to study Zika in North Brazil, I visited several states in North Brazil, and I was able to talk to 91 mothers who had affected children. And 80% of them told me where they felt absolutely nothing when they were infected. So it was very clear that the Zika virus had a tropism toward the brain of the developing fetus. So you knew that Zika virus infect preferential neuroprogenitor cells in the developing brain, inhibiting its proliferation and differentiation. And then we knew that brain tumors are rich in NPCS cells. So we wanted to investigate the outcome of cancer cell lines infected with Zika virus in vitro. So that was our next approach to study Zika virus in stem cells and cancer, and these were the students that were involved in that. And we obtained tumor from brain tumor cell lines, human tumor cell lines. And when we infected these tumor cell lines with Zika virus, we found that there was a high destruction of these tumor cell lines when they were infected with Zika virus in the lab. So there was a decrease in the tumor sphere area and a great destruction. And also we found out that the Zika virus destroyed much more cancer cells than normal neuroprogenitor cells. So then we wanted to test the Zika virus oncolytic effect in embryonic central nervous system cancer cell lines in vivo, which were injected in nude mice. So we injected these nude mice with these central nervous system tumor lines. And after the tumor was established, we injected them with Zika virus. And to our surprise, in 21 of 29 animals, we showed a reduction in the cancer, in the tumor, and in 8 of them, there was complete remission, including metastatic sites. Here you can see how it completely disappeared. And all control animals injected only with the tumor or Zika virus did not survive more than 2 weeks. So this was published in Cancer Research, was the cover Cancer Research, and it received more than 1 million views. But the next question I want to address, how to evaluate Zika virus tropism in other animal models. And we know that dogs can be an ideal model because they can be very similar to humans, not only in sickness, but also in health. We know that dogs can develop gliomas that spontaneously, and they are very similar to human gliomas, so they can provide a very important platform for validating therapeutic strategies in mouse models. So before the pandemic, we were able to identify 3 dogs which have very advanced brain tumors. What are we learning from them? First of all, we injected this virus in the dogs, and we found no negative clinical side effect. This was already published in molecular therapy. But as you know, a video is worth 1,000 words. So I'm going to show you some videos of these injected dogs. This was Mateus, who was very severely affected with very large brain tumor. He did not recognize his owners. He had difficulty in walking. And here you can see him 2 weeks after the virus was injected, already playing with the ball. And here already at home, 1 month before the injection. So here we covered all the clinical and neurological signs after Zika infection. And this was another dog, Nina. She was also severely affected, 12 years old. She could not feed independently. She had difficulty in walking. And again, the outcome after Zika injection, here you can see her at home and running normally. So there is this potential of translational research in dogs in human medicine, and this is a research that we want to continue. We interrupted during the pandemics, but we want to continue. We are starting now new dogs that are going to be injected. So they are very important animal models for human disease because most disease develops spontaneously in dogs, when in mouse they are induced with genetic modification. And dogs share the same environment of about 80% of disease that we see in humans. So we hope that with this will allow translational medicine, because what we found also, which was very interesting, that the virus C can not only destroy the tumor cells, but also it elicit immune response. So this could be very helpful, we hope, to brain tumors, and particular brain tumors that affect children. So despite all the difficulties that we are all facing with the last 2 years, we think that there is light at the end of the tunnel. And I want to thank you very much again for your attention, for the invitation, and to our group in the center, and to all our supporters. Thank you very much. So I'm very happy to answer questions if you have any.
Okay, well, muchísimas gracias, doctora Mayana. Thank you very much for a brilliant lecture. Es importante ahora mencionar si podemos tener un par de preguntas para la doctora Mayana. Vamos a revisar, hay algunas por acá. Permítanme un momento, vamos a revisar el tema de las preguntas. Doctora Mayana. Aquí hay una, what mechanism can be explored to minimize the impact of genetic edition on the epigenetics of individuals?
Well, that's an important question. People are still studying, and that's why we still cannot do this research in embryos that will be implanted, because we still don't know if when we edit a gene, you cannot affect other genes randomly without control. But there are several approaches already to try to study when you do this edited gene, at least in cell lines, to study other genes to see if they are not affected. And this will be very important for safety.
Thank you very much, Dr. Zatz. Bueno, vamos a revisar si hay más preguntas. Perfecto. Bueno, no parece haber más preguntas. Agradecemos de parte de la RELAGH, muy especialmente su colaboración, su tiempo y su experiencia. Y damos un saludo muy especial a la presidencia del ALAG por permitirnos participar en este simposio. Entonces, un saludo fraterno desde la RELAGH. Doctora Mayana Zatz.
Muchísimas gracias.